for today, Dr. Onur Kanisicak. Just a reminder, if you have questions for the speaker, please add them into the Q&A box, and we'll have a Q&A session at the end. So thank you again for joining us, and I'll turn it over.
All right. Thank you, Ron. Thank you for everyone coming today. We have a special guest today to talk about his research, A Gibb. We have known him for a while, but I'll give you if you don't know him yet, let me give you a little bit background of what he did. He got his bachelor degree in Ohio University in 2009, and then he had a little bit of work in between, I believe, but he came back to science, as we like it. And then he got his PhD in 2017 in physiology and biophysics at the University of Louisville with Dr. Brett Hill, working on cardiomyocytes, metabolism, and exercise, and published circulation and several other authorships. And then he moved and started working with his postdoctoral at the John Elrod lab at the Temple University. And since then, he has done some seminal work on both metabolism and fibroblasts and fibrosis in the heart. He has literally cleared the table on awards and speakers and publications. He has published in circulation research, JCI, and got his predoctoral from AHA, F32 from NIH, and recently awarded CDA. Another big thing about him is that he is a great speaker. And he has actually recently cleared the table at the ISHR meetings for the first Young Investigator Award at the North American, and then later on in the World Congress, Outstanding Young Investigator Award for speakers. So today he's going to talk about the glutamine and catabolism and its function in fibroblast. Take it away, Gibb.
Thank you, Onur, for that too-kind introduction. Let me go ahead and get some sharing going here. Okay. You able to see that, Onur?
Yes.
All right. Perfect. Okay. Again, thank you, Onur, and thank you to the JMCC editorial board for the selection of this manuscript for this exciting webinar series opportunity. So, as we all know, heart failure is a clinical manifestation of numerous forms of cardiovascular disease. While there are several important pathophysiological mechanisms that contribute to heart failure, a major one is cardiac fibrosis. Now, fibrosis is the excessive deposition of extracellular matrix, which is a complex meshwork of structural and non-structural proteins. In the heart, this primarily includes collagens, periostin, and fibronectin. Two types of cardiac fibrosis are typically found within the heart. Reactive interstitial, which forms and integrates between the viable cardiomyocytes, is typically seen with hypertension, diabetes, and aging. And you can see this visualized here in these hearts subjected to pressure overload, where collagen is stained in red via Picker Series red. Now, replacement or scarring fibrosis typically occurs when viable cardiomyocytes or tissue are lost due to injury, such as occurs during ischemic insults or, in more severe cases, myocardial infarction. And the fibrosis acts to replace this lost cell area in the heart to maintain structural integrity. However, this accumulation of extracellular matrix profoundly impacts cardiac function, mainly due to a loss of myocardial compliance, or, oops, a little too fast. Loss of myocardial compliance, or, in simpler terms, the ability of the myocardium to contract. Ultimately, this excessive fibrosis accelerates heart failure progression. Therefore, the clinical management of cardiac fibrosis presents a major challenge in improving survival rates in heart failure patients. Importantly illustrated here in a review by Tolquist and Mulkinson, fibrotic initiation and expansion was found to occur from resident cardiac fibroblasts of the TCF21 lineage through elegant lineage tracing experiments. While fibroblasts are always present in the heart, their activation and expansion and pressure overload, shown here 6 weeks post-TAC, can affect myocardial structure and function. In another cardiac fibroblast lineage tracing study actually performed by Onur, they defined that these TCF21 tissue-resident cardiac fibroblasts begin to express periostin, a secreted extracellular matrix protein, upon their stress-induced activation, and that this periostin-positive cell population is necessary for the adaptive healing and fibrosis in the heart. Now, these resident quiescent fibroblasts in the TCF21 lineage are important for cardiac development and homeostasis. In response to stress stimuli, such as profibrotic agonist TGF-beta, these fibroblasts become activated, resulting in migration to the site of injury, a proliferative burst, and the production of ECM components such as periostin, which is an early marker of fibroblast activation. As these stress signals tend to be chronic in cardiovascular disease, these fibroblasts fully differentiate into myofibroblasts, characterized by the de novo synthesis of alpha smooth muscle actin, excessive and unremitting production of ECM components such as collagens, and are resistant to cell death. Interestingly, Onur showed qualitatively that removal of Ang2 pumps from mice was sufficient to reduce alpha SMA staining in the heart, suggesting that it's possible for these myofibroblasts to revert to a less fibrotic state following removal of stress stimuli. This led to the possibility that targeting these signaling pathways mediated myofibroblast activation, differentiation, and persistence may impede or reverse fibrosis. Recent work from our lab identified that profibrotic stimulation results in a rapid upregulation of MICI-1 to reduce mitochondrial calcium uptake through the mitochondrial calcium uniporter. This contributed to changes in cell metabolism, increasing oxidative phosphorylation and aerobic glycolysis simultaneously. Furthermore, we observed an increase in the bioavailability of alpha-ketoglutarate, a cofactor for lysine demethylases, which corresponded with changes in histone methylation and myofibroblast differentiation. However, several questions remained after these studies, such as identifying the primary metabolic substrates, cellular origin of said substrates contributing to myofibroblast formation, and whether these metabolic pathways could be targeted therapeutically to treat fibrotic disease. First, we wanted to really understand how the epigenetic landscape is altered during myofibroblast activation and differentiation. To address this, we isolated adult cardiac fibroblasts from 3 independent mice and differentiated them with TGF-beta for 24 hours. We then subjected these cells to ATAC sequencing, which uses a hyperactive TN5 transposase mutant to cleave accessible regions of the DNA while simultaneously adding adapter sequences, which can then be amplified and sequenced. We performed pairwise analysis with RNA sequencing from the same samples to assess chromatin accessibility and gene transcription changes. From our ATAC sequencing dataset, we observed a global increase in chromatin accessibility 24 hours post-TGF-beta, as indicated here by this volcano plot. Coordinating these changes in chromatin accessibility with changes in gene transcription, 236 of the 254 genes increased in expression were also associated with an increase in accessibility, as one would expect. Interestingly, however, 456 of the 491 genes found to be downregulated were also associated with enhanced accessibility. Conversely, a decrease in accessibility appears to play no role in the context of TGF-beta mediated gene programming, as we observed almost no overlap between these two data sets. In performing gene ontology analysis of these overlapping sets, we found numerous processes that we would typically associate with myofibroblast activation, such as cell adhesion, migration, inflammation, collagen biosynthesis, and organization. So collectively, these observations are important for a couple of reasons. One, this data suggests that while chromatin accessibility is required for TGF-beta mediated increases and decreases in gene transcription, the fibrotic gene program activation and silencing likely lies at the level of DNA and transcription factor binding. Now, metabolism can act as a critical mediator of DNA and transcription factor binding, as various metabolites themselves directly act as cofactors for epigenetic modifiers. For example, alpha-ketoglutarate is a critical cofactor for alpha-ketoglutarate-dependent lysine demethylases, the activity of which we have previously shown to be necessary for TGF-beta-induced myofibroblast formation. Now, while we have previously identified increased alpha-ketoglutarate abundance during myofibroblast formation and that histone demethylation is necessary for the activation of the fibrotic gene program, the metabolic substrates contributing to alpha-ketoglutarate and whether these substrates are necessary for differentiation remained unknown. To determine which substrates contribute to increased alpha-ketoglutarate bioavailability, we utilized isotopic labeled glucose or glutamine to determine how these substrates incorporate into intermediary metabolism during myofibroblast differentiation. Now, briefly, just to describe how this works for anyone unfamiliar with carbon tracing experiments, we can use synthesized carbons with an extra neutron, which significantly increases its overall mass. Now, this increase in mass can be detected via mass spectrometry and allows us to determine where these carbons, for example, from glucose, as shown here, end up in downstream metabolites via the metabolism of that substrate. And importantly, this analysis is only going to provide relative levels of how fractional enrichment and not actual absolute abundances. And so when we visualize this, this is just a representation here, but this graph is representing the entire pool of alpha-ketoglutarate within our sample. For the sake of simplicity, a decrease in the zero isotope log indicated here over time indicates that less alpha-ketoglutarate exists that is not labeled with a heavy carbon. And then this is accompanied by an increase in the other isotope logs or the number of heavy carbons found in a given metabolite. Based on a Timecourse experiment, Timecourse labeling experiment that we did, which I'm not showing here, but is within the manuscript, our results suggested to us that 12 hours of incubation of labeled glucose and 1 hour incubation with labeled glutamine were optimal dynamic labeling times to allow us to assess any potential changes in relative pathway flux following TGF-beta mediated activation and differentiation in our in vitro model system. Fractional enrichment 24 hours post-TGF-beta revealed increased labeling in glycolytic intermediates and downstream end products such as pyruvate and lactate. 24 hours post-TGF-beta indicating increased rates of aerobic glycolysis. No glycolytic intermediates were observed in our labeled glutamine experiments, which would indicate that there's no gluconeogenic capacity of these fibroblasts. Now, while aerobic glycolysis was significantly increased, incubation of fibroblasts with our labeled glucose revealed little to no glucose oxidation in the naive or differentiated state, as indicated by low isotopal log enrichment. When labeled glutamine was provided in a separate set of experiments, however, in as little as 1 hour, we observed a significant increase in labeling of Krebs cycle intermediates, specifically alpha-ketoglutarate, which was enhanced with TGF-beta treatment, indicating increased biosynthesis, which was supported by our measurements of the relative absolute abundances of this same metabolites within our cells. Importantly, this increase in Krebs cycle intermediate biosynthesis occurred at a rate 12 times faster than glucose labeling, indicating the rapidity and likely importance of glutamanolysis in myofibroblast metabolism. Now, in addition to its contributions to alpha-ketoglutarate biosynthesis, there are several possible means by which glutamine could contribute to myofibroblast differentiation. For example, glutamanolysis likely regulates proline and collagen biosynthesis, and these are necessary processes for the synthesis of extracellular matrix components. Additionally, the hexosamine biosynthetic pathway and the cellular redox state have been implicated in myofibroblast formation and the fibrotic phenotype. So therefore, we next asked whether glutamanolysis, or the breakdown of glutamine to eventually alpha-ketoglutarate, is required for myofibroblast formation. To test this, we conditionally deleted GLS1, the first and committed step of glutamanolysis, in fibroblasts containing LOXP sites that flank exon 1. Following addition of adenovirus expressing Cre recombinase in mouse embryonic fibroblasts, we observed a near complete loss in GLS protein expression. Deletion of GLS1 prevented myofibroblast formation, as indicated by reduction in the number of alpha smooth muscle actin positive cells, as you can tell here in our black versus red bars with TGF-beta. Furthermore, while TGF-beta resulted in contraction of collagen gel matrices, black to black, the deletion of GLS1 prevented this phenotype. As glutamine contributes significantly to the biosynthesis of Krebs cycle intermediates, as I just showed, we also assessed whether cellular bioenergetics were altered following loss of GLS protein expression. And while TGF-beta increased both basal and ATP-linked respiration, the deletion of GLS1 in our mouse embryonic fibroblasts prevented this TGF-beta mediated increase, as compared as we compare the red versus the green bars here in this graph. Now, our previous studies on MICU1 focused on cardiac fibrosis. And because of this, we next wanted to ask whether this metabolic mechanism of differentiation was conserved from these more naive mouse embryonic fibroblasts to more primary murine cardiac fibroblasts that would be found in the heart. To test this, we use a highly specific pharmacological inhibitor of glutaminase, CB839, and determine the effects on myofibroblast formation. Pharmacological inhibition of GLS1 significantly decreased both intracellular glutamate and alpha-ketoglutarate levels, while increasing glutamine abundance, confirming GLS1 inhibition by CB839. We found that inhibition of GLS1 prevented myofibroblast formation, as indicated by reduction in collagen 1A1 expression, as well as a decrease in alpha smooth muscle actin positive cell populations, and a decrease in the ability of TGF-beta to stimulate collagen gel matrice contraction. So collectively, these results indicate that glutamanolysis is an essential metabolic pathway mediating myofibroblast activation and formation, and that this pathway may be therapeutically targeted to treat cardiac fibrosis. As we previously shown that alpha-ketoglutarate dependent lysine demethylation is necessary for myofibroblast formation, we next tested whether GLS1 activity is required for the TGF-beta mediated histone demethylation in fibroblasts. Inhibition of GLS1 in primary cardiac fibroblasts was sufficient to prevent TGF-beta mediated histone demethylation, as quantified here from protein immunoblotting. Now, importantly, while GLS1 inhibition, as I just showed, prevents myofibroblast formation, we performed a dimethyl alpha-ketoglutarate rescue experiment, which is a cell-permeable form of alpha-ketoglutarate. And this was sufficient to rescue the myofibroblast phenotype, as we see an increase in the percent population of alpha smooth muscle actin positive cells in the presence of GLS1 inhibition. So these data, in addition to our observed TGF-beta mediated increases in chromatin accessibility that I showed earlier, shed some mechanistic insight into the role of glutamanolysis in myofibroblast formation. Now, in collaboration with Dr. Kent Margulies at UPenn, we assessed the translational potential of blockade of glutamanolysis on the myofibroblast phenotype in cardiac fibroblasts isolated from both non-ischemic cardiomyopathy and hypertrophic cardiomyopathy patients. So in these experiments, human cardiac fibroblasts were co-treated with TGF-beta and our GLS1 inhibitor for 24 hours, after which they were stained for either alpha SMA positivity or assessed for gene program activation via qPCR. And as shown here, inhibition of glutamanolysis was sufficient to prevent the TGF-beta mediated increases in alpha SMA expression and other fibrotic genes, as we compare the red bars to the green bars in all of these graphs. And although we did observe some heterogeneous responses here, for example, here in the gene program between explant number 1 and explant number 2, this is likely due to differences in the disease severity and etiology. And so even though that we do see some heterogeneous responses, the response to GLS1 inhibition on the myofibroblast phenotype generally was conserved across all 3 patients here. So in summary, and this kind of concludes the JMCC manuscript here, but we show that the therapeutic potential of targeting glutamanolysis to prevent myofibroblast formation under stress conditions, and this could provide a novel target to treat fibrosis and heart disease. And so to this point, we next asked, does in vivo targeting of glutamanolysis decrease fibrosis and heart failure progression? So the greatest therapeutic potential of inhibiting glutamanolysis or any interventional strategy lies in the possibility of reversing fibrosis by de-differentiating activated myofibroblasts post-injury under chronic stress stimuli. So we therefore tested if targeting GLS1 in activated fibroblasts in vivo would lessen fibrosis and improve cardiac function in a mouse model of pressure overload heart failure. So to test this, we crossed our GLS1 flox mice with the periostin inducible Cree line developed by the Mulkinson lab and a Cree-dependent fluorescent reporter line to both label and temporally delete GLS1 exclusively in activated myofibroblasts post-differentiation. Following 16 weeks of TAC, FACS-sorted TD tomato positive cells confirmed GLS1 deletion via immunoblotting. Assessment of the peak aortic pressure differences and of the total number of TD tomato positive cells isolated from these hearts revealed no differences between our two genotypes. And so this really helps to show that we had equal stress between both genotypes here. Loss of glutamanolysis in activated myofibroblasts, however, revealed the preservation of contractile function starting at the 10-week time point and on, as indicated by our measurements of ejection fraction here by echocardiography. Further characterization of the changes in cardiac function using B-mode speckle tracking at the 16-week post-TAC time point revealed increased radial strain rates during systole, indicating improved contractility. Importantly, we also observed an increase in the rate of relaxation indicated by the inverse radial strain rate, which is calculated during diastole. And so this finding really is important because diastolic dysfunction is highly correlated with tissue fibrosis. To this point, we assessed collagen deposition via picroceros red, which revealed a significant increase in our TAC control animals, which was prevented following loss of glutamanolysis in these activated myofibroblasts. Importantly, collagen deposition was similar between groups at an earlier time point, here 6 weeks post-TAC, confirming our hypothesis that fibrotic regression occurs following loss of glutamanolysis in these activated myofibroblasts, and that this really is reversing disease progression. So again, we assess the translational potential of blockade of glutamanolysis on myofibroblast reversion in cardiac fibroblasts isolated from our patient explants. In these experiments, human cardiac fibroblasts were maintained under chronic TGF-beta stimulation for 48 hours with glutamanolysis intervention at the 24-hour mark. These cells were then stained for alpha-smooth muscle actin positivity and assessed for fibrotic gene program activation, again via PCR. And as you can see here, inhibition of glutamanolysis was sufficient to revert these myofibroblasts back to a less fibrotic phenotype, as indicated by our red bars versus our orange bars in both of these patient explanted cardiac fibroblasts. Now, collectively, these results indicate the therapeutic potential of our metabolic intervention in not only preventing, but reversing cardiac fibrosis in cardiovascular disease. So this leads us into the last question proposed at the start, which is what I'm currently pursuing in the lab, which is what is the source of glutamine utilized by these fibroblasts? And is fuel availability a regulatory mechanism in fibrosis, or is it merely sufficient? In a recent metabolomic study by our collaborator Zolta Rainey, arterial coronary sinus differences in human heart failure revealed that of all the metabolites secreted by the human failing heart, glutamine was the highest. And so additional evidence from Heinrich Tegmeyer actually concluded that in the perfused working heart, transfer of carbons from glutamate to glutamine does occur, yet the reverse process was undetectable, suggesting that in the adult heart in cardiomyocytes, there's very little uptake and breakdown of glutamine. RNA-seq analysis of mouse hearts subjected to TAC or MI in our lab revealed a significant increase in the expression of nearly all glutamine transporters indicated here, and in GLUL, specifically in our TAC model. And this is the gene that encodes for glutamine synthetase. So these results here suggest the possibility of enhanced cardiac glutamine production and secretion in heart failure. And so to directly test the role of glutamine synthetase in cardiac pathophysiology, We generated a cardiomyocyte-specific tamoxifen-inducible knockout model of glutamine synthetase and subjected these mice to pressure overload-induced heart failure. Loss of cardiomyocyte glutamine synthesis preserved LV structure and function, indicated here by fractional shortening measurements, and prevented fibrotic infiltration within the left ventricle. And so these results suggest to us that there is a potential for a cardiomyocyte to fibroblast metabolic crosstalk mechanism that mediates fibrosis and cardiac health. Lastly, we asked whether targeting glutamine synthetase after disease onset would rescue or mitigate heart failure progression. In this study, we waited 8 weeks post-TAC prior to tamoxifen administration to induce cardiomyocyte-specific deletion of GS. Again, aortic pressure differences were no different between our groups, indicating a similar stimuli. And as you can tell here by our short axis fractional shortening measurements or our long axis volumetric ejection fraction measurements, that these mice track very nicely up to this 8-week time point as far as their decline in function goes. But following administration of tamoxifen after 8 weeks, these groups do seem to separate. And we do seem to observe a preservation of cardiac function following loss of glutamine synthetase in the cardiomyocytes. And so now we're currently working to further phenotype these cohorts and have experiments planned to demonstrate directly that cardiomyocyte-derived glutamine is contributing to myofibroblast formation, as well as exploring the cardiomyocyte-specific protective effects of loss of glutamine synthetase in heart failure. And so, in summary, myofibroblast differentiation is associated with these dynamic changes in chromatin accessibility, primarily increased accessibility, which correlates with activation of the fibrotic gene program and with our metabolic reprogramming that we observe. Glutamine metabolism is rapidly increased during fibroblast activation and is the primary carbon source for the biosynthesis of the epigenetic modifying cofactor alpha-ketoglutarate, as well as for the biosynthetic anabolic reactions such as collagen biosynthesis and for ATP supply, which all supports differentiation and eventual tissue fibrosis. Pharmacological inhibition and genetic disruption prevents myofibroblast and even reverses myofibroblast formation. And this phenomenon is recapitulated in fibroblasts from the human heart failure. And if we target GLS1 in vivo in these already activated fibroblasts, we can improve and mitigate cardiac functional decline following pressure overload. And so lastly, cardiomyocyte-derived glutamine, we feel could be a signal or a fuel that activates these fibroblasts to initiate fibrosis, as indicated by our GS deletion and cardiomyocyte studies. And now we're really trying to further expand on this by really trying to expand our understanding on metabolism and how this contributes to fibroblast biology and fibroblast differentiation in heart failure. And here's just a quick graphical summary of these conclusions, which is constantly changing as we continue to work towards our full understanding of how metabolism directly contributes to cardiac disease progression and other possible targets that we can use to promote cardiac health and improve outcomes in disease. And so with that, I'd like to thank everyone in the Elrod lab, specifically Dr. Elrod, my postdoc mentor, individuals of the CTM here at Temple who have been very helpful with these studies as well, collaborators at the University of Louisville, UPenn, and UCLA. I'd like to thank the ISHR and American Heart Association, NIH, for the funding that I've received and for the opportunity to present at a lot of the ISHR conferences most recently. So with that, I thank you all for listening and welcome any questions.
Thank you, Andrew. This was awesome. I love the fact that you actually give the big picture, combining your papers together.
Thank you.
While we're waiting for others to chime in with questions, I have a couple for you. I guess we can do a private Q&A session the whole rest of the day, but let me start with a couple of the important burning questions in my head. So far, you looked at the fibrotic aspect of, fibroblasts and what they do with disease, and clearly with this epigenetic changes. But have you looked at other crosstalk with other cell types in your setting? I don't know how much inflammatory crosstalk you'll see in the TAC model. Maybe you're going into the failure stage, maybe you'll see something. But for example, with MI model, there's a huge flux of immune cells, and there's a lot of crosstalk between fibroblast and immune cells, right? And if you prevent this differentiation, which you carefully call myofibroblast, so you're kind of specifically talking about the ECM aspect, maybe. But can you, comment on whether you have seen any changes of this crosstalk cytokine expression of fibroblasts, and how that can affect the physiology?
Certainly. So we haven't gone to, the crosstalk mechanism now that we're looking at, right, is the cardiomyocytes communicating with the fibroblasts to promote their differentiation. Now, infiltrative cells such as macrophages or these immune cells can obviously activate fibroblasts themselves, right, through the production and secretion of TGF-beta or other inflammatory cytokines that can bind to various receptors on fibroblasts and promote their differentiation. Lactate is another signal for fibroblast activation and differentiation as well, another metabolic signal, which we would think would be enhanced during cardiac disease as glycolysis is increased and oxidative phosphorylation becomes compromised. And these macrophages and immune cells are highly glycolytic as well. So I think that there is certainly multiple metabolites of interest that could be contributing to myofibroblast activation and things like that. And I am certain that there is things secreted from the fibroblasts or myofibroblasts themselves that are then acting to either dampen cardiomyocyte function, or to enhance or promote the anti-inflammatory or pro-inflammatory phenotype within the heart.
Right. Next phase of the question maybe is probably out of your scope, but have you done this developmentally? Have you knocked it out with a, I don't know, global fibroblast or, TCF21 maybe, post-birth, like neonatal mice, and look at the development of the heart? How much of this, metabolic activation pathway affects the growth of the heart?
So we haven't done any developmental studies, but there are some studies, right, where deletion of PDGFR alpha positive cells, or at least there's some evidence, right, that if you delete at least that proportion of fibroblast cells within the heart embryonically, that the heart's developed fine, and there doesn't appear to be any developmental issues if you delete all these fibroblasts from the heart from birth. Which is very interesting, if that is true.
Yeah. So exactly. So the question in hand, I guess, is we know that fibroblasts and cardiomyocytes communicate a lot in disease states, and we assume that there's a critical part of that in homeostatic too. But if you look at Cinicinati's work from children's, they have also knocked out the periostin expressing cells, the activated fibroblasts during that so-called regenerative phase of the mouse heart, and then they've seen a reduction in the cardiomyocytes with proliferative markers, right? So there's something going on that I'm wondering, and I think you're referring to Michelle's recent paper with the PDGF alpha knockout. I'm not sure how much developmentally they got into it, but I'm very curious, is this energetics and metabolics of the fibroblast are the key to growth phases as well, or not? You know what I mean?
Certainly, yeah. So we did some proliferation. We did a proliferation assay on these cells when we inhibit glutaminase. And they do proliferate a little bit slower, but it's not, it's not significant. Like, it's not crazy that they proliferate 50% slower or anything. It was like 10% slower. So there's still other mechanisms that allow these cells to proliferate. Now, what's interesting is that, for instance, if you increase glycolysis in a fibroblast, either, genetically or, well, specifically genetically, that this also promotes their differentiation. However, we see here that if we delete or pharmacologically inhibit glutamanolysis, that doesn't affect glycolysis in these cells in response to TGF-beta. So TGF-beta increases glycolysis, and glycolysis remains increased with inhibition of glutamanolysis. So to me, that says that, while increased glycolysis is sufficient, it's not necessary, right? And that I think the link between glutamanolysis and glycolysis is what's really key. And it seems that it's really this glutamine utilization that is driving the activation of these cells in the heart.
Yeah, I like that you're saying it's probably what's physiological versus artificial, right? I mean, if you start fibroblasts, they also activate. Like, there's so many things we can stimulate these guys and activate, but what's happening in vivo physiologically is what you're referring.
Exactly.
Here.
Yes.
Right.
Yes.
So there is one question. What are the other pathways may get activated by pharmacological inhibition of glutamanolysis?
So, like, as I said, we know that, so we kind of saw this intermittently, but it appears that if you inhibit glutamanolysis, that you do get an increase in glycolysis. And that's probably just to, fuel ATP production, fuel nucleotide and amino acid biosynthesis, lipid synthesis, in order to maintain the proliferative capacity of these cells. So we do know that glycolysis is typically activated. And we do know that with TGF-beta, we saw increases in several amino acids that were synthesized by our stable isotope tracing. And we did see nucleotides as well being increased. And so, it's the metabolism stuff is certainly complicated, because if you inhibit one pathway, a lot of other pathways, are likely to change. And, we unfortunately aren't able to inhibit and look at every single pathway. But we do know that glycolysis is increased, and that at least oxidative phosphorylation is prevented, and that amino acid synthesis is reduced with glutamanolysis as well.
Maybe there's another question coming up, but it got short, so I'll wait for that. Ron has a question.
I have a question. So
Certainly.
I can wait. It looks like the Q&A question just came on, so I'll ask mine after this one.
Okay, so the question is, I'm dyslexic, so I might butcher it, but I'll try my best. It's elementary reading skills here. So what is the major energy substrate in fibroblasts, like fatty acids in cardiomyocytes?
So it seems that at least the major energy substrate, at least one of them is glutamine. It's a primary one, because specifically, if we give TGF-beta, oxygen consumption rates are increased, and this is directly tied to ATP production basally. And if we inhibit glutamine utilization, that completely prevents that increase. Now, the cells also use a ton of glucose, and there's going to be a ton of ATP that's generated from aerobic glycolysis. However, because we're seeing that these changes, because these cells are still oxidative, right? So they're not completely losing the ability to become oxidative phosphorylation or generate ATP in the mitochondria. But this seems to be independent of the ability of the cells to activate the fibrotic gene program, which is in the nucleus and involves the transcriptional machinery there. So at least, in our culture conditions, we don't have fatty acids on board. So we know that they're not using fatty acids, at least in our in vitro system. They do use pyruvate, and they do uptake, there's some literature that they do uptake lactate. and can use that as a fuel source for again, both energy, but then also for synthesis of various amino acids and things like that. And I think there might be some literature out there. I think I saw one paper where they might even utilize some branch chain amino acids.
Ron?
So I guess my question has to do with more the fibroblast contribution to the metabolic milieu of the heart as an organ. So, I think your work really uncovered something unexpected, that fibroblasts are central players in metabolism. But what I'm trying to wrap my head around is just on the per cell basis, and other than really advanced injury, fibroblasts are not a large component of what makes up the heart. And so do you think in, let's say, an infarct or in an area of fibrosis in the tissue, are there subdomains of metabolic alteration? So I guess, in other words, how does the fibroblast, or does the fibroblast as a cell type change the metabolic profile of the heart overall? Or do you think it's, the myocytes in an area of fibrosis have an altered metabolic profile compared to the rest of the myocytes?
That is very interesting to think about in that capacity. So we know that these fibroblasts, at least, that are kind of more interstitial, right, are within and between the cardiomyocytes, and could kind of be farther away from any capillaries or blood supply, right? So within those little, I guess we can call them like fibroblast niches or something, right? They could be really dependent or reliant on anything that is secreted by the cardiomyocytes. And so that's kind of where we think this glutamine secretion by the cardiomyocytes and the utilization by the fibroblasts, where that really lies and has impact. Because glutamine in the circulation is the highest amino acid in circulation. It's 200 to 600 micromolar. And so it's really not limiting. But if these fibroblasts are away from that source, or if the cardiomyocytes really are releasing excessive amounts of glutamine, those local concentrations could be much higher. And then we know that the fibroblasts are highly aerobically glycolytic and release a lot of lactate. And the fibroblasts could be using that lactate as an additional fuel source, potentially, to help try and drive as much ATP production as possible and try and limit the energetic deficit that's going on. But as far as, testing these things are very, very difficult, as you can imagine. So I'd have to really think about how we would be able to test metabolites coming from the fibroblasts that could be influencing the metabolism of a cardiomyocyte. And maybe it's as simple as, deletion studies of cardiac fibroblasts from the heart entirely. But then you're going to probably run into other issues that you can't really then tease out exactly what's going on.
Coming back to that regionality, I have a question about the model you used in your previous paper with the TAC model. So you show the interstitial fibrosis kind of goes away a little bit, right? A couple questions, right? Number one, did you see a reduction in number of fibroblasts active at that time with the fluorescent reporter you used? That's
so we did.
Yeah. Go ahead, finish that. Yeah.
So it didn't seem like we saw a reduction, at least based on our fact sorting shown here between our two groups. So we don't think that, with this deletion system, that we're getting a less of a fibrotic response because we have less activation.
Right. Okay.
So we don't think it's that. And the half-life of GLS1 in vivo is proposed to be about, I want to say it's 5.1 or so days. So that's going to be about 21 days roughly before it's completely gone.
Mm-hmm.
And, with the TAC models, all these cells are going to come out at various different times. So it's not just going to be like an MI, a large insult of fibrosis. This is very gradual and gradual accumulation of fibrosis, which is why we think that, we don't really see a change here until later. change in function here until later in our model system, because we think that at this point, somewhere in this 6 to 8 to 10 week range is where we're getting optimal fibroblast activation, we're getting optimal fibrosis, and then we're getting the start of optimal deletion of GLS1 and fibrotic reversion, which is supported here by our hydroxyproline assay results that at the 6 week, we really don't see any difference in fibrotic accumulation. But then after that point and later in disease, when we start to see that functional change, that we do see the fibrotic regression.
Right. The second phase of this is of the interest of different subpopulation of fibroblasts, if you believe in them or not. But do you see the perivascular fibrotic response in the TAC model that you're using? Does it disappear as much as the interstitial one?
So we haven't looked specifically at that, but I know that, and I think it might be from you potentially, is that the periostin expression doesn't really correlate too well with the perivascular fibroblasts. I think you had a poster at that a couple years ago. And so what we're trying to really understand is these different cell populations and what's really occurring at the local versus the non-local. One thing that I would think is that, especially within our glutamine synthetase deletion model, is that, as I mentioned, since the circulating levels of glutamine are so high, the perivascular fibroblasts could likely not be affected by the deletion of GS in our cardiomyocytes. And so we are looking to try and do not only histological analysis of that, but then also some single cell seq analysis to see how does deletion of GS in the cardiomyocytes affect the populations, and is it only interstitial versus perivascular, or is it all around?
All right. Yeah, we can go on and on, but Ron, I think we can wrap this up. Otherwise, it's going to turn into like another PhD thesis exam.
Sounds good.
All right. Thank you, Andrew. Always a pleasure to see your work. And
thanks, guys.
we'll be looking out for your next publications.
Awesome. Thanks so much.
Thank you, everyone, for joining today. Have a great day.
Thanks.